recording is in progress. Note that we'll be recording the session. Yeah.
So I can start screen sharing. Is that okay?
Sure. You can do that. So good afternoon, everyone. So welcome back to the CBC virtual seminar series. So we are very happy today to have Dr. Maria Babak from the University of Hong Kong to give us a talk today. So Dr. Maria Babak is an assistant professor at the City University of Hong Kong and a PI at the Drug Discovery Lab. So she completed her Ph.D. in bioinorganic chemistry at the University of Vienna back in 2014, mentored by Professor Bernhard Keppler and Professor Christian Hartinger. So back in 2015, from 2015 to 2020, so Dr. Babak worked as a postdoc research fellow at NUS, right? Mentored by Professor Ang Wee Han, where she developed a true passion for drug discovery and target identification. So subsequently, Dr. Babak completed the high-impact cancer research program at Harvard Medical School in Boston. Her research interests lie at the interface of chemistry and biology and medicine, and focus on the discovery and preclinical development of anti-cancer drugs for resistant and aggressive cancers with limited treatment options. So these are what we call high-value products, difficult to achieve targets. That's very interesting. So for today's talk, she will talk about the development of metal-based drugs for treatment of undruggable cancer. Let me talk a little bit about the abstract. So despite significant progress in the field of cancer, some cancer types are still being characterized by very short life expectancy. Understanding of biological vulnerabilities of cancer cells open new avenues for treatment of those cancers that are yet to be drugged. So in this seminar, she will talk about various strategies of targeting drug-resistant cancer cells with metallodrugs with unconventional modes of action. The discovery of their unconventional mechanism of action provides a promising route overcoming drug resistance and tumor relapse. So with that, let's welcome Masha. Masha, to you, please.
Thank you very much, Ken. Thank you for your introduction. And thank you everyone for joining the seminar today. And I would like to start saying that I spent a very significant part of my life, of my adult life in Singapore. And I really love Singapore. And I think I am one part of my heart is Singaporean. So I moved to Hong Kong from Singapore in the middle of pandemics, not so long ago, approximately 1 year ago. And I can tell you what people miss the most about Singapore after moving away from it is, of course, the food. I am so sad that I cannot eat Singapore food. And especially surprisingly, I miss bak kut teh the most, which was not my most favorite dish when I lived there, but now it's the one that I miss the most. And I really hope that the borders will be open soon and I will go back to Singapore because I really love it. And now I would like to speak a little bit about my research. And as you probably understood from my name, Masha, and from my accent, which is quite strong, I am Russian. And I was studying in Moscow, did my undergrad in Moscow, and then I moved to Lille, France, and this was even before I started my PhD. And then after that, I was doing my PhD at the University of Vienna in Austria. And Vienna is an amazing city, not as amazing as Singapore for me, but it's still very amazing. And it's famous for its culture, it's famous for its coffee, and I spent quite a nice time there until my supervisor decided to move to New Zealand, and I followed him there. And this was a completely different experience because New Zealand is a very nature-oriented place, and the type of work there, how people work, it's much more relaxed. But it was an interesting experience. And then I moved to Singapore and I worked in NUS under the wing of Professor Ang Bihan, who is an amazing mentor. And I also spent 1.5 years in Boston, also in parallel with Singapore. And I was in Harvard Medical School, where I actually learned cancer from the perspective of doctors. My background is a chemistry, so I'm a chemist, but I tried to learn also molecular biology and a little bit of oncology when I was in Harvard. And eventually I moved to Hong Kong, and this is where I am right now. And another thing I miss about Singapore is that the weather doesn't change, because now we're at the moment when it will become cold. And even though I'm Russian and I have a winter background, I don't like when it's cold, and I would prefer that it's hot all the time. So very soon winter will start. So anyway, let's stop talking about how good Singapore is, and let's speak about research. So my first part of my talk is targeting cancer stem cells. And before I start about talking about cancer stem cells, I would like to share with you one very personal information about me, which is I'm a very big Beatles lover. And my favorite Beatles member is George Harrison, and he is famous for his songs, of course, but also, sadly, he is famous for the fact that he was diagnosed with cancer 3 times in a row. And his first cancer was throat cancer, which is already pretty bad, especially for a singer, and he was diagnosed in 1998. And after that, he went through the surgery and chemotherapy, and he was quite successfully treated. And after that, he came back to his music and to his playing. And you can actually, if you are also a Beatles lover like me, you will be able to understand the difference in his music, where he became very philosophical and India oriented. And then in 2000, he was diagnosed with cancer again, with lung cancer. And this is where it became much more serious, but still he was treated and he was still okay. And unfortunately, in 2001, very shortly after the second cancer was fully treated, he developed the third one. And this time it was a very deadly brain cancer. And probably you know that as soon as there is a brain malignancy, then it's much more difficult to treat because of blood-brain barrier, and in general how our brain behaves. It doesn't like anything foreign in it, and that's why, unfortunately, George Harrison died. So why I tell you all that is because when the doctor says cancer, or nowadays, this is not a death sentence, especially if a patient comes to a doctor at a very early diagnosed stage, and then it's not a problem. The drugs, they work very well, and patients can live happy life after that. And when the real problems occur, when cancer comes back, and especially when cancer comes back in a metastatic way, so that means the primary cancer goes into the distant sites of the body. So that means that the real problems is the actual cancer recurrence rather than cancer itself. And here on this graph, you can see the effectiveness of treatments of patients with metastasis. And the first line of treatment is when the patients were not treated with anything else. And you see that the efficacy is very good. So when we look at doxorubicin or paclitaxel, the efficacy is 50%, which is pretty good. Of course, not 100%, but it's good. And when it's the second line of treatment, then the efficacy drops significantly, and especially shockingly, for 5-fluorouracil, there is almost no efficacy at all. So that shows you that if the patient is here at this bars, then he or she is okay. But then the second time, it's already a much bigger problem. So what to do then? The researchers, of course, started to think about it, how to deal with this problem. And there are a lot of groups all over the world, including ours, who deal with this kind of issues. And there was this kind of theory developed several years ago. It's not new, maybe more than 10 years ago even, which is called cancer stem cell theory. And what it says that the tumor is the heterogeneous population of different cancer cells. And first of all, we know that cancer, it's not one disease. It's a combination of many different cancer diseases. But even within one type of cancer, the tumor is not a heterogeneous population of bad cancer cells. It actually has different types of cancer subpopulation. So according to this cancer stem cell theory, the tumor is the heterogeneous mass. Let me take my pointer. Okay, here's my spotlight. So this is the heterogeneous population, and we have one small quiescent subpopulation of cells, which is actually responsible for high tumorigenicity of tumors. So that means that when we treat the tumor with conventional chemo, the bulk of the tumor is removed, whereas the quiescent very bad subpopulation of cancer stem cells is left there. And this is the one that is responsible for tumor relapse, according to this theory. So what does it mean? That means that we remove the part of the tumor that is relatively easy to remove. However, that part that is difficult to remove, we actually even concentrate it. So that means the tumor becomes even more aggressive. So that means if we use, let's say, cancer stem cell targeted therapy, then we can specifically selectively remove the cancer stem cell subpopulation, and then the bulk of tumor will either be removed by normal chemo or can be also removed by this cancer specific drugs, cancer stem cell specific drugs. So that's the idea. Now, let me remind you what stem cells are. So stem cells, they're basically very boring cells. If we look at this picture, we see very beautiful nerve cells, cardiac cells, blood cells, and then we have this very round, boring cells, which basically do nothing. But in reality, they don't do nothing. They do very important things. So they, first of all, regenerate into themselves. And second, they regenerate into something else that the body needs. So what I always say that if we have an injury and our bike has an injury, then the bike is probably gone if it's beyond the repair. But if it's our bone or if it's our skin, then it can be repaired exactly because we have stem cells in our body. And that's why when you become old, then it's very difficult to repair this kind of injuries because there is basically not much stem cells left. So the same concept works for cancer stem cells. So cancer stem cells, again, they only have two functions. They either regenerate into themselves or they regenerate into different types of differentiated cells. But because this is a bad guy, this will differentiate into cancer cells, so differentiated cancer cells. So that means that main two functions of the cell is self-renewal and differentiation. The properties of cancer stem cells are highly tumorigenic. What does it mean? That actually means, and you can easily see this in the lab if you know how to work with mice. So for normal cells, if you inject the cancer cells in the mouse, you need hundreds of thousands of cells for tumor to grow. However, if you inject the cancer stem cells, you need less than 10 cells. This is not even hundreds of cells, less than 10 cells you inject into the mouse, and the tumor will grow very aggressively if you inject 10 cancer stem cells. So this is a quiescent cell population. That means that it's not active, it's sleeping. And that's why it's also very small. So that's why it's very, very difficult to detect it. However, scientists are smart, biologists, they managed to find the distinct cancer stem cell markers for each subpopulation of cancer cells. So let's say breast cancer cells, cancer stem cells, colon cancer stem cells. So they found the specific markers. And for example, for breast cancer stem cells, we have the subpopulation CD24 minus CD44 plus and LDH plus. There are a lot of other different markers. So then this brings us to the question topic. So if cancer stem cells are highly resistant to standard chemotherapy, then how are we going to target them? Because I'm a chemist, right? As a chemist, medicinal chemist, I'm interested in developing the drugs that will target this specific particular population of cancer cells. And that means clearly this should not be the drug that targets the same cancer pathway as standard chemotherapy, like cisplatin or oxaliplatin or paclitaxel. So this will be the compounds that target alternative cancer pathways. And how do we find this kind of compounds? Of course, we cannot do it without biologists. So initially, biologists have to find out what is the vulnerability of these cancer stem cells. And then we can try to find the compounds that will target this vulnerability. So the good model for breast cancer stem cells is the triple negative breast cancer. So here you see the range of different cancer cell lines, starting from luminal breast cancer MCF7, which is a hormone-dependent cancer, all the way to triple negative breast cancer MDM-B231. And here in red, you have endothelial cells. So why I'm showing you this picture? Because you can see just from the morphology of the cells that the hormone-dependent breast cancers, they have very round shape, round morphology. And when we move to the more aggressive types of cancer, they become more elongated and more similar to endothelial cells. So that means that the vascular mimicry is increasing when we go from luminal to basal, and also the occurrence of metastasis increase as well. So what is interesting for triple negative breast cancer, the first site of distant metastasis is actually brain. And this is very bad because you can imagine some types of metastasis, for example, bone metastasis or lung metastasis, doctors know somehow how to treat more or less efficiently. But if the first site of metastasis is brain, then it's a pretty bad thing, right? So triple negative breast cancer is called triple negative because it doesn't have any of the receptors that other types of breast cancer have, for example, estrogen, progesterone, and EGFR. And that means that this type of cancer cannot be treated with standard hormonal or targeted therapy, right? It cannot be treated with HER targeted compounds, or it cannot be treated with hormones for estrogen or progesterone receptors. So interestingly, triple negative breast cancer responds well to chemo, because obviously it cannot respond to anything else. So this is where the chemist can actually celebrate. It responds to chemo, but patients, they come to the doctor, they get chemo, they respond very, very well, but very, very quickly this cancer develops a resistance. Therefore, at the moment, there is no actually defined chemotherapeutic scheme for it. So what we actually developed in our lab, and this was developed in NUS in the lab of Professor Ang. So they found out that there is this 3-component assembly. This is the reaction where we can in situ produce a large number of ruthenium complexes with a very high yield and high purity in situ. So we don't even need to isolate the compound. Of course, when you are synthesizing your drug candidate has to be exceptionally pure, otherwise it will never go into any clinical trials. But as a screening method, if you have the purity more than 85%, this gives you a very good model to screen a large number of compounds. So the big advantage of inorganic chemistry is that compounds have very nice colors. And when usually we do the chromatography, if we can do chromatography, then our compounds have exceptionally beautiful colors. They can be yellow, they can be red, they can be pink, they can be blue, purple, anything you like. And this is one of the things I like in inorganic chemistry in comparison with organic. So if you look at this compounds here we have the 96 well plates, and in each well of the 96 well plate, we have a different compound, which is synthesized via this combinatorial assembly. So that means this ligand, and this ligand will complex, they will be all different. That means we can create a very, very large number of combinations. And in each well, we have a different type of ruthenium complex. So we can synthesize as many as we like, depending on how many precursors we can get. Then we choose the model. What will be the model for our breast cancer stem cells? And we choose the model of breast cancer spheroids, in particular MDMB231 mammospheres. So mammospheres means breast spheroids. So these spheroids grow in non-attachment conditions, and why we prefer them to breast monolayer is because that they have higher population of breast cancer stem cells in it, and that's exactly what we want. So as I told you, the characteristical surface markers of breast cancer stem cells are CD24 plus and CD44 plus and CD24 minus. So we take 2 different types of mammospheres, and then we measure CD44. Here you have green color. I don't know whether you can see it very well. Here we have red color, which is for CD24. So that means MCF7 mammosphere, they get the completely different opposite surface phenotype, which we do not want. Whereas MDA, they are exactly what we need. Very little of CD24 and CD44 is positive. So we use this breast cancer spheroids. We grow them in the non-attachment conditions in 96 well plates. And then after that, we add our huge libraries of ruthenium compounds. And for each of them, we measure the cytotoxicity. So here you see the concentration at which your drug kills 50% of the cells. And you see we have a very big range of them. So we call this series routines or routines, and we change the substituent here, or we change the substituent on the arene ligand of the sandwich complex, half sandwich complex. So here you see if the cytotoxicity is higher than 20, then it's not very interesting for us because we're looking for very good drugs. So probably those compounds might still have some kind of activity, but we, because we have hundreds and hundreds of them, we choose only those that are probably the best. So we found that these compounds that have low micromolar cytotoxicity, possibly if we continue testing more, we would find even better compounds. But at this moment, we decided to go with this four. So one of them is very active. If we replace the arene here from triisopropylbenzene to hexamethylbenzene, then the cytotoxicity changes significantly. It actually drops if we have this ligand. And in this case, we have different substituent. We have chloride here. So when I say chloride, this is chloride here. When I say hexamethylbenzene, this is arene here. So don't mix them. So here we have the drop of activity as well. And this is very interesting. Why do we actually get such a drop of cytotoxicity if we only change this part of the molecule? Very, very interesting. Very small change, but very huge difference on breast cancer cells. So then, of course, as I told you, it's extremely important to synthesize very, very pure complexes. That's why we isolated these complexes separately, and we made sure that we get crystalline structure. Here you can see for two of them, we have an X-ray. Here we also got X-ray later on, and then we made sure that the purity of these complexes is more than 95 or even 97%. And then we also made sure that this complex are stable and we can proceed with other biological testing. So here you see the cytotoxicity table. We tested it in MCF7 and MDA, and for MDA we used monolayer and we used spheroids as well, so determine their cytotoxicity. And you see that for monolayer the activity is actually pretty good. It's the low micromolar range. So in comparison, we use cisplatin and oxaliplatin, which are standard anti-cancer drugs, and they are usually not active for breast cancer, for triple-negative breast cancer. And this is what we see here. We see that there is no activity. Actually, for those of you who don't know this kind of studies, I have to specifically explain that if the number is high, that means the activity of the compound is low. But if the number is low, that means the activity is high. So that means if you have 0.2 and 27, 27 is not good, the compound is not toxic, whereas 0.28 means it's very good because your compound will kill cells at less than 1 micromolar concentration. Also, in addition, we compared our compounds with the stem cell selective compounds that were identified via the screening of huge NIH library in MIT, and they found these three compounds, and then we used them for comparison. And what we see here, we see that salinomycin, which was the best compound from that screen, it shows us very similar cytotoxicity, but our compound is still better, both in monolayer and the spheroid situation. So then, as I told you, it's a very, very important property of cancer stem cells is to regenerate and become themselves. So if we, let's say, we kill our cells, but some of them are still alive, then they can regenerate into themselves again, and then there will be no point in doing what we are doing, right? So we have to make sure that we are actually inhibiting their self-renewal capacity. So to do that, we do this experiment where we grow the mammospheres, we add the drug, and then we see how their size or number changes. So don't try to understand this graph. It looks pretty difficult for quick understanding, but here you see the picture and you can understand this picture very well. So here you have the control spheroid, the picture of one spheroid. So when we add cisplatin or oxaliplatin, which we know have no effect on triple-negative breast cancer cells, especially cancer stem cells, the size of the spheroids do not change, also even becomes higher. For salinomycin, for our positive control, we have the decrease of the spheroids. And then when we look at our drugs, this is our least potent drug. And these drugs basically break all the spheroids into very, very small pieces. Now you can tell me that what does it matter if these compounds break the spheroids into pieces, if these pieces are still very viable? And you will be completely right if you ask me this question. So that's why we reseeded this residual cells into new petri dishes, into the same non-attachment conditions. And then we saw whether the cells can continue, proliferate, and grow without anything else. and we do not add any additional drug. We just see whether there will be any secondary mammospheres growing. And what we see for our least active drug, indeed, they start growing. For salinomycin and control, they start growing. And here we see, yes, to some extent, they still grow, but to a very, very small extent. Then we also check the surface markers. This can be done by flow cytometry, and I'm not really going to go too much into details because probably most of you are not biologists, and it will be too difficult to understand. But what is important here, that we have the significant decrease of the surface marker, which is responsible for cancer stem cells. What does it mean? Our cell, if it is a cancer stem cell, it has the particular handle which can be recognized, the surface marker. So if we have many cells, we will have a lot of this subpopulation of the surface markers. So the fact that we do not see anything here means that we probably have cancer cells, but they are not cancer stem cells, which is very, very important. So even if we still have some cancer cells there, they are not cancer stem cells. And that means we can break them with or kill them with standard chemotherapy rather than this very specific drugs, right? Then what is very interesting is to check whether our cells become less aggressive. It is very good if we can kill the cells to some extent. Maybe we can also decrease the markers. But if the cells, residual cells, are super aggressive, then they will still be able to metastasize, and that's not what we want. So here you have a very specific microfluidic device, and here you have the zoom of this part of this microfluidic chip. So here, this is the channel where we put collagen, and we embed our spheroids in it. So they are alive, they are just stuck in a collagen. On the two sides here, we have the nutrients. So these nutrients cannot go inside collagen, but at the same time, spheroids that are embedded inside, they can feel that there is food here, and food here. So because they're embedded in collagen, and collagen is like a glue, right? So the cells try to move through this glue in the direction of the food, otherwise they will die. And this is what you can measure. You can measure the direction, the radius and diameter of how far your cells manage to go through the collagen in the direction of food. And that's how you can identify how invasive your cells are. So we do the proof of concept first. We take the very highly invasive cells, and we have non-invasive MCF7 cells. And then we see these are our spheroids at 0.0, so before we start the experiment, and this is after 24 hours. And you see, amazingly, these cells don't care about the food. They just stay happily where they are because they're not very invasive ones. But this, however, you see it breaks into small pieces and starts moving in the direction, all possible directions, just to make sure that the food is there. And so that means this is the right model, and then we continue doing it with the drug. So we do exactly the same experiment, but we start injecting here the media with the drug. And what we see that for control cells, they are going everywhere they want, but for our drug, they stay where they are. So that means, you see, for this type of cells, they stay with or without drug. They don't want to move anywhere. But these cells, without the drug, they move where they want. But if you add the drug, then they stay where they are. And salinomycin more or less does the same, although if you see 28 hours, it already kind of started to move. Okay, and it's also very important that these cells are not dead. Of course, if they are dead, they will not move anywhere, but they are alive, they're just hungry. So what we see here, we see that our drug, the lead drug candidate, decreased the invasive capacity significantly. It was actually the best one. Of course, if we increase the concentration of the drug, then we would have even better results. But our point here was to use a very low concentration of the drug that will not kill the cells, but will show whether they can stop their invasive potential or not. So this is all very good, but what is very important is that we have some results in mice, right? Because sometimes we have a drug that works very well in cells, but then we move it to mice and nothing is happening. And then, of course, if you move it to a patient, then especially nothing will happen. So that's why it's very important to spend money and do the animal experiments. So because we are working with breast cancer, it is very important to do the very realistic model, the model where mice develop breast cancer. So what we do here, we do some kind of mouse surgery. We're opening the mouse where the nipple is. So obviously mice, they don't have breast, but they have a lot of nipples on them. So we are opening the skin, we expose one nipple, and then we inject breast cancer cells inside, and then we do the suture. So it's like a surgery on a mouse. And then these tumors start growing on the nipple. So it's a very, very realistic breast cancer model. Now, these cells that we are injecting, they are the same MDA and B231 cells. So they are human cells, and we inject them into a mouse. So the mouse would not like it, obviously. That's why they have to be immunodeficient. How can we monitor this tumor if the tumor is inside under the skin? We will not be able to see it, right? So that's why we are transfecting our cancer cells with the luminescent tag and then luciferase tag, and then we can do live cell imaging. So in fact, the mice that you see here, they are not dead. They are happily sleeping under anesthesia. And here we have a cone that is attached to the mouse nose, and then we can measure our signal. The more red the signal is, the bigger is the tumor. And then at the end point, we also isolate the organs and we see whether there is any signal in the organs. The good news is that our drug decreased the size of the tumors in the mice significantly, and we proved that it does it exactly the same way in mice-like cells. The bad news is, if you look at the organs, we do not see any kind of signal there, both in control mice and in drug-treated mice. And that means that we cannot monitor the anti-metastatic activity of our drug simply because even our control mice do not develop it. So we did not manage to create the model where we can get metastasis. It is possible. It's just probably we need to use a different cell line. So then we check the toxicity, and here we managed to see the biodistribution of our drug, and we see that there is nothing in the brain, which is a good thing because it does not cross blood-brain barrier. There will be no neurotoxicity, and there is quite a lot in kidney, spleen, and liver. This is pretty normal for this type of drugs because these organs are excretion organs, and they don't like any kind of drug in their body, so they try to excrete it. Of course, if we have a high concentration of the drug in the organs, this can be related to the toxicity of these organs. So that's why we did the standard test for toxicity. This here are the enzymes of kidney and liver, and we see that there is no statistical difference between them. So there is outlier here, but more or less, it's more or less the same. And statistically significant difference was not detected. So this one I will skip. I will go to animals again. So of course, we were very unhappy that we did not manage to see any metastasis because this was the whole point of our experiment. And there is another model. There is a model of hepatocellular carcinoma, which is also a very, very bad cancer. So triple negative breast cancer is probably the main cause of death for women who die from cancer, and HCC is one of the main cause of death for men. Very common, both of them in Hong Kong and in Singapore. And they are very highly aggressive and resistant, and can be treated more or less efficiently with these two pills, but chemo is more or less not working on this drug, on this tumor. So here we do the same animal model, but it's a little bit more complicated because if we just inject the cancer cells in the liver, they usually do not pick up. So the tumors do not pick up and they don't start growing. So you need to do even a little bit more complicated type of animal surgery where you take the piece of tumor, you chop it into small pieces, and then you graft and implant it into the animal liver, and then you do the suture and you close the mouth. The same principle, our cells are transfected with the luciferase tag. That's why we can monitor the growth. So here we see that relative tumor burden is significantly decreasing. However, what is very important, finally, we got our metastasis. And here you see the size of the tumors is huge. So this was actually right before the end point, because if you want to do humane experiments, you need to be sure that your animals do not suffer, do not feel any pain. That's why after this moment, we actually sacrifice them. And what you can see, we see that their lungs are full of metastasis. You see a lot of metastatic nodules there. And then if you decrease if you add your drug, you see, this mouse is totally healthy. It does not have cancer at all. So it did have cancer, but then the cancer disappeared. This mouse also, nothing, no metastasis. This mouse, nothing. This mouse, nothing. So this is an extremely good result. You have an extremely invasive liver cancer. You see your mice are almost dying. And then you have your drug and you see them free of cancer. So this is really, really amazing. Then we tried to do some mechanistic studies. Since most of you are not biologists, I'm not going to go through details. And this is where this project is right now. So we are trying to understand what is actually the pathway that is affected by this drug. So now we still have time. So let me move to another part of my talk. And Ken, when we are running out of time, please let me know so we have enough time for Q&A. So sure. My second part is targeting cancer cell metabolism. And here I would like to start my talk with showing you all these books that I found in Google. So this is very fashionable right now, the ketogenic diet. Everyone is doing keto. Everyone is losing weight on keto. And so these authors, they decided to treat cancer with keto diet. If you look at these books, everyone is downloading them. Everyone is buying them. People say that you can cure cancer with this, but of course it is not true. They are actually not good people who sell this kind of books. So if you think that people who are buying this kind of books, they are not smart, you will be wrong. So here we have probably one of the smartest people in our world who unfortunately died already, and this is Steve Jobs. And Steve Jobs, he was diagnosed with pancreatic cancer, and he was one of the most firmest believers of ketogenic diet, and even rather than ketogenic diet, he was believing in fasting, veganism, and drinking raw juices. So when he was diagnosed with pancreatic cancer, instead of behaving very quickly and reacting quickly and going through surgery, which is not always possible with pancreatic cancer, and also going through chemotherapy, he was extremely against that, and he started to fast. Because he was saying that there is a research, there is a scientific research that cancer cells, they like sugar, and if we deprive them from sugar, then there will be no cancer. So he was doing it for 9 months. For 9 months of his last months of life, he was drinking raw juices and was actually starving himself. So as you know, during cancer, people lose a lot of weight. This is the side effect of having this disease. So in turn, for Steve Jobs, he actually starved himself even more, and he was really like, you can see this picture, which probably he wouldn't like to be public. So why would such smart people like Steve Jobs believe in this kind of nonsense? Because there is a very famous review. Every scientist working with cancer knows it. And this is the review that says that all the cancer cells, they have something in common. And this is called hallmarks of cancer. And these hallmarks of cancer are the properties of each and every cancer cell. For example, unlimited proliferation, ability to metastasize, evade apoptosis, and so on. And here you see one particular hallmark, which is the reprogramming of energy metabolism. So this is known as Warburg effect, and it was discovered by Nobel laureate Otto Warburg. And basically what Otto Warburg found, he found that cancer cells, they love sugar. Okay, so cancer cells, they love sugar much more than normal cells love sugar. And that's what all these people use when they write their books, that cancer cells love sugar. This is true. This is written in a Cell journal. And that's why if we deprive them from sugar, that's where they will die. But it's much more complicated than that, of course. So not only the cancer cells love more sugar, they also process it differently. So if we look at how the sugar is processed in normal life, in our bodies, we have the sugar in presence of oxygen, it becomes CO2. And if there is no oxygen, then it forms lactate. And we all know this burning feeling after gym or jogging. And this is what happens when the sugar is converted into lactate. And this happens in normal cells only when there is no oxygen. And This process here is much, much less efficient. So only 2 ATPs formed during this process and 36 ATP molecules is formed during this process. So interestingly, for normal cells, they are going through the very, very energy efficient process, and they go through this production of 36 molecules of ATP. However, when it happens in cancer cells, doesn't matter if it's in presence of oxygen or absence of oxygen, they still go through this process, which is very energy inefficient. And then you will think that this is very stupid of cancer cells, right? Because they want to have more energy. Why would they go through a less energy efficient process? The truth is it's much faster. And for cancer cells, it's always the question of speed. They want to develop faster. They want to grow faster. They don't have time to go through a slow, even more energy efficient process. So let me just go into this, and I will try to sum up everything. So our favorite form of sugar is called glucose, and there is a very, very, very difficult pathway here, and medical students in medical universities, they learn this in details, Krebs cycle, but luckily we don't need to do it now. The only thing we need to know is that glucose, it's at some point becomes pyruvate, and this is a very important moment because pyruvate either can go to lactate or it can go to CO2. And for normal cells, this is what happens when there is no oxygen, and this is what happens when there is oxygen. So there is a catch. There is a pyruvate dehydrogenase that is actually responsible for going this direction. However, pyruvate dehydrogenase kinase is forcing the cells to go into this direction. And of course, cancer cells are smart. They overexpress this kinase. That's why they force their glucose to go into this production. So the idea is that we can inhibit this kinase, and then this production will go normal way. So that's what we were trying to do. We were trying to create the, let me show you. We were trying to create the platinum-4 prodrug with this kind of specific inhibitors, and we wanted to do two-stage targeting. On the first stage, we will selectively target cancer cells rather than healthy tissue by using nanoparticles, because nanoparticles, they are using this very specific phenomenon, which is called EPR, enhanced permeability and retention effect. And because of this phenomena, if you have larger particles, they might be stuck inside the blood vessels and they might work longer. Whereas if you have small molecules, they will go in and out, and then they might not work well enough. So that was our first stage of targeting. And the second stage of targeting was the mitochondria targeting. So we put the special mitochondria targeting pendant in order for our cells not only to go selectively in tumor, but also into selectively into mitochondria. So let me finish, let me show you this one last slide, because I know that everyone likes to see the in vivo studies. So here we see this very cute mouse. This mouse has a hair, so that means it has immune system. So that means we're actually injecting the normal mouse cells, mouse cancer cells into a mouse, and mouse grow their mouse tumor. And here we see there is no effect when it's only PBS. When we use cisplatin, there is some effect. When we use our drug without nanoparticle encapsulation, there is better effect. If we increase the dose, even better effect. But if we have our two-stage targeting system, the tumors don't even develop. And this is an amazing result, because look at the difference between here and here. Here we have 1.5 centimeter the area of the tumor. It's like this, it's huge. So the mouse is carrying this kind of tumor, whereas here it doesn't even develop. So that's how effective your drug is. So I guess this is where I can stop now. Should I stop now and then we can have the Q&A?
Sure, we can do it. We can.
Okay,
stop.
because I have much more slides, but I think that it's enough. So let me just show you my very last slide. I want to show you my team. So we are a very new developed team, and we are a drug discovery lab. So we are specifically working with drugs, and this can be not necessarily anti-cancer drugs. They can be some immune modulating drugs, or if we have interest in other types of diseases, we can develop drugs for them as well, or screen them. So the that's why we have a small chemistry group, and we also have the molecular biologist, and we are lucky to have doctors on our team who can do this kind of complicated animal studies with the organ analysis. And if anyone in NTU is interested to join us, we have the deadline for PhD applications every 1st of December. And yeah, we welcome everyone in our group if you are interested. Thank you very much.
Thank you. Thank you so much. Thank you. Yeah. So thank you, Masha, for the talk. And I would like to open up the Q&A to the audience. So please briefly introduce yourself. And before you post your questions, please unmute and feel free to ask some questions, please.
Hello, Maria.
Hi.
Thank you for your presentation. May I ask how you do the mode of action study?
Mode of action study?
Yeah.
Yes. This is a very long answer to this, but let me just briefly tell you. For example, for these compounds, right? We first analyze whether this compound is going into the cell or not. So the biggest advantage of our drugs is that they have a metal center in it. So that's why we can use the special technique, which is called ICP-MS, inductively coupled plasma mass spectrometry, and it allows us detection of the drugs at very, very low level at any kind of biological sample or organ or bodily fluids, even feces, so we can really track where our drug is. So first of all, we check whether our drug is inside the cells at all. If it is, then we can try to see the DNA damage. Then we can try to see the apoptosis. So there are several markers that are responsible for it. And then we can do flow cytometry. We can do Western blotting. We can do confocal microscopy. And this is a good question how we did it, because I actually skipped that part. So for the first part of my talk, we had to decide first what is the vulnerability of cancer stem cells. And biologists found out that this is the sensitivity to endoplasmic reticulum stress. So endoplasmic reticulum is a very specific organelle that is responsible for folding of the misfolded proteins. And apparently cancer stem cells really don't like when something is happening not in their way in this organelle. So that's why we were trying to see whether our drugs do it. And indeed, they could induce some kind of stress in this organelle. And that's how we could show it. And this can be done either by flow cytometry or Western blotting, for example.
Thank you. And so through what you illustrated before, so you can localize it, target the mitochondria, right?
Yes. So,
may I whether will you load the docking site on the mitochondria?
Yeah, so that's exactly what we did. First of all, you can separate your organelles using special kits, because there are special fractionation kits. And we separated them into 3 parts. Here is nuclei, mitochondria, and cytosol. So then you can use this technique that I just told you, I inductively coupled plasma mass spectrometry. So you digest each fraction separately using nitric acid, and then you detect the amount of metal in it. And then you can compare it, nuclei, mitochondria, and cytosol. So for example, here, if you look at nuclei and mitochondria, so just one second. This is our cisplatin, and this is our lead drug candidate. You see that in nuclei, their amount is the same. However, if we compare the mitochondria, this is our cisplatin, and this is our lead drug candidate. It is significantly better accumulated inside mitochondria, as expected.
Thank you for your illustration. Thank you so much.
Yeah, thank you. Thank you, Langsing. So on this topic, yeah, Masha, I'm just curious. Does CSC, cancer stem cells, has endoplasmic reticulum, has ER organelle inside the cells?
Oh, okay, so endoplasmic reticulum is the organelle that exists in every cell. In all our body, we have endoplasmic reticulum because this is responsible for protein folding. So proteins, they are very big structures, and if they're not properly folded, there will be simply no space in the cells for them. So endoplasmic reticulum is responsible for it. So there is one way to kill the cell if you create such amount of stress for it that it actually cannot fold it properly anymore. And then it's either doesn't fold it at all, or it misfolded, so it's like very messy. So cells don't like it, and apparently this was discovered in MIT already 10 years ago. So the cancer stem cells, they are particularly vulnerable to this kind of event. So if you do the DNA damage, they can find a way. If you induce the reactive oxygen species, they can find a way. But if you mess with the endoplasmic reticulum, then they die.
Okay, so if there's a way to selectively work on the ER stress, there will be a very interesting could be a very interesting outcome from there. Yeah.
Yeah.
Yeah. Okay. Just a quick question on the PDK. So there are four PDK, four different types of PDK. So are you working on a pan inhibitor concept, or you're targeting one of them?
To be honest, I didn't even know that there are 4 PDK. I just, yeah, this is, I learned something new today. So the idea was that there is this organic PDK inhibitors, dichloroacetic acid and phenylbutyric acid, and they actually target different parts of PDK, but which PDK, I honestly don't know. And the problem with these drugs that even though they are effective, you need huge doses for it. And this is where you need a chemist, because if the chemist looks at the structure, you will immediately understand that this molecule is quite a bad one because it does not go through the membrane. So that's why you need gram scale of this molecule to have some kind of effect. And that's why we modified it with better metal fragment, and then it became much better.
Yeah, thank you. Oh, I know that there's more than one type of PDK. I used to be a medicinal chemist in the local drug discovery company, and PDK was one of the targets that we are working on. Yeah.
There is a question in the chat.
Yeah.
In the chat. So the question is, what are the advantages of this metal complex drugs as compared to conventional small molecule drugs or biologics? Okay, this is the question which my students get every examination from every examiner. Why are you working with metal-based drugs? So thank you for this question. So first, let me tell you what can be the disadvantage of the metals. Even so many years after cisplatin was discovered by Bernhard Rosenberg, people still think that metals are very bad for them because of their toxicity. And this is true. Sometimes these molecules can be very toxic. However, what people forget that organic drugs nowadays, very toxic ones, cytotoxic ones, they are also very toxic. So here we can really discuss what is more toxic. In terms of advantages, here you should understand a little bit of coordination chemistry. So coordination chemistry tells us that metals can have various number of geometries and also coordination numbers. And what does it mean? That means that we can easily, okay, sometimes not easily, but most of the time easily fine-tune the molecule and to make it have the properties that we want. For example, if we want to add some molecules to increase lipophilicity, we can do it. If we want to increase the water solubility, we can do it. If you are a very good organic chemist, probably you can do it as well. But I can tell you that the level of difficulty in coordination chemistry is way, way lower. So it is really easy because of such a wide range of geometries and coordination numbers. You can basically attach what you want. You just need to think of the structure and you will be able to do it. So probably this easy fine-tuning of the properties is the biggest advantage of the drugs. Now, in terms of pharmacokinetics, unfortunately, not so many groups are at the stage where they are measuring the pharmacokinetics. So I can't really tell you much about it, but I know for those very, very advanced drugs, I can tell you one example, for example, why metal-based drugs are better. So I'm coming from Vienna, and my professor, Ph.D. professor, is very famous for one drug. It's called Keppler. So this drug is a ruthenium drug, and its cytotoxicity is very low. So the IC50 is like around 100 micromolar. If it was in 2021, this drug would never even get into a good journal, okay? But because it was developed in 1990, the professor believed in it, and he pushed it into clinical trials. So what happened? Nowadays, there are patients that live only early because they are on this ruthenium drug. And not only they leave because of it, but they also have no side effects, which is amazing. They don't have hair loss, they don't have vomiting, they don't have nausea. The only side effects that they get is that their blood becomes green, and but it's transient, it's not permanent, so eventually it becomes red again, and then of course patients are happy because they leave and they also don't get significant toxicity. So this is the advantage of metal-based drugs.
So there's a related question. What about the cost? The cost of inorganic drugs, is it more expensive?
So because metal-based drugs sometimes are very easy to synthesize. So for organic ones, you have the whole range, you know, like a multi-step organic synthesis, and then eventually you might actually have, I don't know, 1 milligram from 1 gram, right? So for metal-based drugs, you might actually save the cost on this. So it depends on which metal you are talking about. So for example, copper. Copper is super cheap. So if you find a good copper drug, then you will have no problems. Gold is expensive. But then again, you always should understand that if you have like very complicated molecule like paclitaxel, to synthesize it, it might be even more expensive than the cost of gold.
Yeah. So there's another question on the, what do you think the factors, what are the factors of the lead drug contribute to the specific mitochondria targeting?
I'm not sure what you mean by factors, but if we look at the drug, 1 second. Here, this drug is targeting mitochondria because it has the special targeting pendant. So this molecule is called TPP. It's triphenylphosphine moiety, and it's known to bring the molecule to the mitochondria. So actually, if you remove it, then your molecule will go less into mitochondria. If you replace it with, let's say, ER targeting handle, then it will go to ER. But for whatever reasons, it's much more difficult to bring the molecule into other organelles. So it's easier to mitochondria. Maybe it's more developed, more studied. I don't know.
Sure. Thank you. So is there any other questions?
Hi, Dr. Masha. I'm curious, you say about this delivery vehicle is the nanoparticle, right, in the system?
Yes.
So how do you control the specificity of the nanoparticle into the cancer cell? That's the first question. Like maybe you put some ligand or some modification on the surface of the particles. And then the second question, how do you control the release of your drug from the nanoparticle so that it is able to go into the mito?
Thank you. Some nanoparticle specialist here. So most probably you know this much better than me. So first question is about, second question about release. So we did the experiments where we used the what's it called, dialysis membrane, and then we have our drug within the nanoparticle, and then we see how much is actually in the solution. How can we measure it? Again, our magic method, ICP-MS. We had our own ICP-MS in the lab, so all these kind of experiments we could do easily. And the first question was whether it's a specific nanoparticle with any targeting ligand. So I am not a specialist in nanoparticles, so basically we chose the easiest one. And how we confirm the specificity, you can't really do it in vitro, you have to do it in mice. So because we actually have this here. Here you see the no, I don't have it here. So what I wanted to say that if we see the very significant increased effect when we have our drug, so this is our drug and this is our drug in the nanoparticle, then just by seeing the effect, we say that it's more specific. But we did not do any targeting molecule on the nanoparticle. Can be done, actually, this TPP moiety can be on nanoparticle and not on the drug, also possible.
Okay, thank you.
Thank you.
So on this particular graph, is it IV or PO?
No, we never did PO. We always did, it's not even IV, here it's IP. And the reason is simple, because it's the simplest to do. So we started with IP, just injecting in the peritoneal cavity.
Okay, thank you. Okay, I think we have some time. We have a bit more time for one last question.
Hi, hello again.
Hi.
Hello. May I ask, because I think your target is the triple negative breast cancer cell, right? But you're using nanoparticle to transport your drug. So would it mean your drug can target other cancer cells?
Okay, so first of all, these are two different projects. One project was the first one for breast, specifically for triple negative breast cancer, and there we did not use the nanoparticle. And this one is more, we developed the concept, so we did not specifically do it for particular type of cancer. So we actually, this model is just a colon mouse cancer. Now, whether our drug can be used on different types of cancers, this is an extremely good question. And unfortunately, I don't have an answer, because the cancers that we tested, we saw that, yes, there is an effect there. But to make sure, to give the good answer, we need to screen on many different cancer cell lines, and then we can say, okay, here it's active and here it's not. But I believe that because all drugs have mitochondria, then it should work on different types of cancers.
Thank you.
Jiren, you have one question?
Oh, sorry. Okay, so I was just recently, hi, Professor Masha. I was looking at your recent paper in Anguante about the metformin prodrug. So I was just curious, because you mentioned it was very easy to functionalize this sort of inorganic complex, metal complex for to control the release of your drug. So I was wondering, looking at the structure of the metformin prodrug, addition of a simple ketone functionality on the bridging carbon seems to confer like much better, much lower stability, which in return allows for a more controlled release of metformin as compared to the other two analogs that you have tried. Is there some sort of rationalization behind it?
Okay, so first of all, thank you for reading my papers. It's very nice to hear that. And second, usually the standard rationalization for this kind of work doesn't matter what kind of metal drug we use. We are trying to prepare a series, and whereas for organic compounds, sometimes it's very easy to create a series where you have methyl, ethyl group, phenyl, and so on. For metal-based compounds, it's not always possible, because, for example, for these gold complexes that you are talking about, we have metformin, and we don't want to modify it because this is the drug we want to bring into the cells. So then we can only modify the cyclometylated portion. So then we are trying to make it to see what we can do with it. So for example, to make it more lipophilic or less lipophilic. So this has been already done before in another work where not we, but our collaborators, they introduced the long pendants, and they saw that with the increase of lipophilicity, the compounds became more toxic. But in our case, we tried to change it. It's hard for people to understand because they don't have the structure. So we tried to add that ketone and see whether it will affect the stability of the complex, because the idea was to trigger the release of metformin. But for this particular complex, I have to tell you, it's not very easy to synthesize because it's a very temperamental complex. So I see here is my student is in the audience, and he is exactly now trying to do it. I can tell you he is not very happy.
All right, I see.
Okay.
Okay.
Thank you. Thank you so much. And I should like to conclude the Q&A session and therefore conclude the seminar. Thank you once again, Masha, for taking your precious time to attend this.
Thank you, Ken.
Yeah, we are at the same time zone, so it's not that bad. So I will be following up with you for some potential collaboration.
Of course.
Yeah, because we recently found some interesting compounds that we can target lipid droplets and ER selectively. So maybe we can try to work on it. But it's organic compounds, not inorganic compounds.
That's okay. The biggest problem with organic compounds is that you cannot use my favorite ICP-MS method. I'm so comfortable with it, and once I get organic chems, then what to do? HPLC, UVVs, then it's I'm very stressed. But it's okay.
Or you can use LCMS, MS method.
Yes,
yeah,
yes.
it's always available. All right. Okay, so thank you so much. And for the rest, especially students, please record our attendance by referring to my latest message there. And I'll have to share one. I will have to share the next screen.
Yep.
Yeah. Okay, so once again, please join me in thanking Dr. Maria Babak, and thank you so much. So please take care, and I hope to really see you in a physical person again. Thank you.
Yes, thank you. Bye bye.
Bring you to your favorite
bakute.
Yes.
Yes. Okay, bye bye.
Bye bye. Thank you.